Hey, Hammy here, back with the last video for Chapter 3 over Section 3.5, in which we will look at nucleic acids. The two main nucleic acids that we will discuss are DNA and RNA, and hence the terms you'll recognize the N and the A from nucleic acid, deoxyribonucleic acid. Okay, the DNA, that's our genetic material. Going to store all the information on how to make those proteins that we talked about in the last video, in what order to put all those amino acids. RNA has a wide range of functions, from protein synthesis to gene regulation, and some even act like enzymes. Okay, the monomer of DNA, RNA, of nucleic acids is a nucleotide, and we'll take a closer look at structure here in a second. But remember, there's three things: a phosphate group, a pentose sugar, and a nitrogenous base, or nitrogen-containing base. Remember, we usually use a circle for the phosphate group if we're just drawing rough shapes here. Pentagon for the 5-carbon sugar, or the pentose. Sometimes you'll just see me write 5C, that's pentose or 5-carbon sugar. And then you've got some kind of nitrogenous base. I usually use different shapes kind of like this, but there's some are ring structure, single ring, some are double ring structures. The 5 types of nitrogen, or the 5 nitrogenous bases found are A for adenine, found in both DNA and RNA, guanine found in both, cytosine found in both, and then thymine, which is only in DNA, and uracil, which is only in RNA. So that's one of the differences between DNA and RNA. Nucleotides are joined together by a series. Again, how do we join biomolecules? Dehydration synthesis, which forms a linear molecule. And we'll look at pictures of these in future slides of this sugar phosphate backbone with the nitrogenous bases kind of stick off the inside or towards the middle. This slide just contains a lot of the sort of molecular structure of these three parts. Again, you see the phosphate group, which we normally just draw as a circle, as a phosphate with a bunch of oxygens around it. So again, you can see kind of why we would draw it as a circular shape. The pentose sugar, you've got two, DNA versus RNA, the deoxyribose versus the ribose. Okay, where does that name come from? Look, right here, there's an OH group. Here's just a hydrogen. So it is without an oxygen, or deoxygenated, or deoxyribose. Okay, so it's a ribose sugar without the oxygen. And now you see why we get that in the name. The nitrogenous bases we split into pyrimidines and purines, whether they are single ringed or double ringed. I always remember the single ring, the smaller rings are the bigger name, pyrimidine. Whereas the double rings, the bigger molecules, have the smaller name. So you have cytosine, thymine, uracil, all belong in the pyrimidine category, whereas adenine and guanine belong in the purine category. Okay, the backbone of the nucleic acid is composed of alternating sugar phosphate. Okay, so you got a sugar, phosphate, and then your nitrogenous base off the inside. And then that will connect again to a sugar, or to a phosphate, to the next sugar. Okay, we call this right here the phosphodiester bond. Phosphodiester, because it's between the sugar and the phosphate group. And that just keeps linking back and forth, back and forth, back and forth. Okay, and that sort of makes up the backbone. And RNA would just be that single strand with the nitrogenous bases off the inside, whereas DNA is double-stranded. And we'll see here in a second that their hydrogen bonds will hold the adenine and the thymine together, and the cytosine and the guanine together. This was figured out because when they did analysis of DNA of different critters, we found that the amount of adenine always equaled the amount of thymine in a species, and it varies from species to species. and the amount of G always equaled the amount of C. It wasn't always 25%, 25%. It wasn't 1/4 of each. These could vary some, but A and T always were about the same percentage, and G and C were always the same percentage. And that's how they sort of figured out that, hey, probably wherever there's an A, there's a T. So if they're always bonding together, there's equal amounts of them. And obviously, if you add A and G, always equals the number of T and C. Just some basic math there. This chart just compares the differences between DNA and RNA. Just, I guess, 4 main differences. One is the sugar, deoxyribose versus ribose, which we looked at. The 4 bases, A, G, T, and C in DNA. RNA has A, G, C, and U. Uracil replaces thymine in DNA. DNA is double-stranded, which kind of twists around itself, that double helix, whereas RNA is just a single-stranded nucleotide or nucleic acid. So DNA, again, is in a double helix. RNA is just single-stranded. The first slide here, just taking a look at RNA structure. Again, notice single strand. Here's our phosphate, sugar, phosphate, ribose, phosphate, ribose, phosphate, ribose backbone with the nitrogenous bases sticking off of one side. So here you can see guanine, which is a purine, uracil, pyrimidine, adenine, which is a purine, and cytosine, which is a pyrimidine. In DNA structure, you can actually see that now this is a double helix. This is always kind of cool here, the space filling model. Remember the molecular sets, if you would actually build this thing, how it would look instead of just geometric shapes. But you notice here's the sugar, phosphate, phosphate, sugar, phosphate, sugar, phosphate, sugar backbone down each side. And then the nitrogenous bases form what we call the rungs. Think of climbing up the rungs of a ladder. They're the rungs down the middle. And you see these dashed lines between cytosine and guanine that always pair together. There are 3 hydrogen bonds. And between adenine and thymine, there are 2 hydrogen bonds. Okay, and that just helps keep that helix stable. Remember, DNA helicase can come down through there and unzip those when we're doing DNA replication and when we're copying the DNA. The other nucleic acid that we talk a lot about in biology is adenosine triphosphate, ATP. ATP is a nucleotide composed of adenine and a ribose sugar. Okay, those together, adenine and ribose, that's where we get adenose. Adenine, ribose, adenose. So adenosine, and then it's got 3 phosphates, hence the triphosphate. And we'll look at the structure on the next slide. ATP is a high-energy molecule. It's formed between that second and third phosphate group. So we say the terminal phosphate bond. So between that second and third phosphate, when that's put together and broken apart, we put a little bit of our mitochondria, put a little bit of energy in to put it together. And then when it's broken apart, it releases some energy. So it's this gaining and losing of an inorganic phosphate. Sometimes you'll see it represented by P with a subscript I for inorganic phosphate. Sometimes I will just draw a P with a circle around it, just indicating that phosphate group. The hydrolysis of ATP can be coupled with chemically favorable reactions in the cell to allow the reactions to proceed. So as we're breaking down carbohydrates over here, it's releasing energy. That's going to be put into ATP. And then ATP will go over to things like nerve impulses, pumping ions or active transport out of a cell, muscle contraction, and it will release that energy for those functions to happen. And then it'll come back to the energy releasing reactions that are taking place. So they're often coupled or paired with energy absorbing and energy releasing reactions in the body. Therefore, we call it the energy currency. Just like we use currency, paper, money, coins, and we trade back and forth to buy goods. The energy, ATP, is what gets the energy and then gives it off, gets the energy, gives it off to keep our cells running. This is just, again, the space filling model of ATP right here. You remember right here is the ribose sugar, which we usually use as draw as a pentagon. Up here would be the nitrogenous base, okay, which would be, in this case, adenine. So you can kind of see why we use the shapes that we do. So up here's adenine. Here is ribose. Remember, those together make adenosine. And then here's our phosphate. The yellow represents the phosphate group, phosphate group, phosphate group. Remember, phosphate groups have these oxygens hanging off the outside of them. So we have 1, 2, 3 phosphate groups. It is this bond right here, the terminal phosphate bond between the second and third phosphate, that is often broke apart, and you're left with ADP, adenosine diphosphate, an inorganic phosphate group, often dubbed P subscript I, and our little burst over here of energy. Okay. Notice the double arrows. This can go either way. Notice the water. This is a dehydration, synthesis, and hydrolysis reaction, depending on which way these arrows go. Okay, that's the end of chapter 3 and all the major biomolecules that we'll talk about here in AP Biology this year. Go back, review them. If you have questions, write them down so that you can ask me in class. All right, this is Ham out.